Okay, so hello. I'm going to discuss today activity number 12. It will focus on the Papa Nicolo staining method. So, the staining method is actually considered to be a method of choice under cytology. So, in cytology po kasi, we are dealing here with highly specific and accurate techniques for us to differentiate tumors na related lang po sa inflammation or tumors related lang po sa degenerative diseases o di kaya tumors na, anong tawag nito, malignant. Okay, so usually yung mga techniques under cytology na dyan nagfo-focus to differentiate tumors that could be inflammatory, that could be caused by degenerative diseases, or it could be from malignancy. So the advantages of the techniques used under cytology, such as your Pap staining method, is of course, first, the easiness. The easiness of obtaining the material, the easiness of obtaining the specimen, to be exact. Okay? And actually, this easiness that I am talking about actually led to a significant reduction of the incidence of cervical cancer. Kasi since madali lang mag-collect ng specimen, so madali lang i-perform yung madali lang ma-perform yung test, so madali lang ma-diagnose yung mga babaeng may cervical cancer. Okay? So, I have already mentioned that your Pap staining method is the well-known and the most commonly used technique under cytology. So, let us now discuss that one. So, your Pap smear is again a test in gynecological cytology. It is used for detection and prevention of cervical cancer. And that's what I have mentioned a while ago as well. Na yung mga techniques under cytology, may mga techniques tayo under cytology, na nag-allow ng mga tao to diagnose cervical cancer. Okay? So, the question now is, how was it developed? So when we talk about the question, how was it developed, we need to look back as to how this method was established. So this one, this test, was actually developed by George Papanicolo. I'm not sure how to pronounce this name kasi foreign ang name niya. So dito lang muna tayo sa English translation ng kanyang name. So George Papanicolo. Okay. So, paano niya na-establish yung ating Pap smear, yung ating Pap test? So, he tried to correlate the sexual cycle of women with the cellular patterns by examining the vaginal secretions of both rodents and humans. It is also during this time that he also tried, since meron naman siyang specimen, meron siyang vaginal secretions, he also tried to identify stages of exfoliated squamous epithelial cells in terms of maturation. And also, as a consequence of that one, he was able to differentiate the malignant cells as well. Okay? So, because of this development made by George Papanicolo, that is why he was considered to be, or he is considered to be the father of modern cytology. Okay? So, let's now describe Pap test. Okay, or your Pap smear, or your Pap staining method. So this one, this test actually involves a multi-chromatic staining of your cytological smears. So when we say multi-chromatic, multi means many. So ano po marami dito? Chromatic, from the word chrome. Maraming colors na involved. So ibig sabihin po, kapag nag-stain kayo gamit itong method na ito, ang end result niya, makikita nyo sa smear, iba't ibang kulay. Marami pong colors na makikita nyo po dito. And it is a very reliable technique. Okay? So, if you want to know more about the advantages of Pap smear staining method, you can refer to your lecture, kasi I think i-discuss yan siya, or you can read in your book. It's actually stated in page. Sasabihin ko na yung page para Madali lang malocate. No? It's actually on page 312 of your book. So you can see there the advantages, no, of Pap smear staining method. Okay? So we have mentioned a while ago that this method is actually a good technique, no, under gynecological cytology. But actually, class, no, aside from the vaginal smears or from the vaginal secretions, this technique could also be utilized in other types of specimens as well. Okay? So, any specimen po na nag-i-involve ng cells, no? So, cytology man ito. So, nag-i-involve ng cells, you can actually utilize, no, Pap smear staining. Okay, so what are the other specimens? So aside from the gynecological smears, you can also use Pap staining method when you are when you want to observe, no, the cells in the sputum, in the brushings and washings, in the urine, in the cerebrospinal fluid, in the seminal fluid, no? In the serous, synovial fluids. Your FNA, no? FNA means fine needle aspirate. So you can use Papa-Nicolau staining for that one. The tumor touch samples and other samples, or rather other specimen types, no, na nagko-contain ng cells. Okay? So now, your Papa-Nicolau stain, class, especially the classical form of the Papa-Nicolau stain, actually involves 5 dyes in 3 solutions. So let me begin with the 3 solutions. So what are these 3 solutions I am referring to? So the 3 solutions I'm referring to here is your hematoxylin, your OG6 or your orange G counter stain, and your EA counter stain or your eosine azure counter stain. So these are the 3 solutions I'm referring to a while ago. How about for the 5 dyes? Kasi po sinabi ko diba kanina na when we talk about the classical form of Pap stain, it involves 5 dyes in 3 solutions. So I've already mentioned the 3 solutions. So now I will mention the 5 dyes. Okay? So the 5 dyes that we're referring to here are still your hematoxylin, your orange G, and then you have your eosine Y, your light green SF yellowish, and your Bismarck brown Y. So these are the 5 dyes that I'm referring to. Okay, so let us describe each. Let's begin with your hematoxylin. So again, don't forget when we talk about hematoxylin, that is a nuclear stain. Okay, and it is said to be an unmordanted hematine, or rather, it contains an unmordanted hematine. So what do we mean when we say unmordanted hematine? Unahin po natin define yung hematine. When we say hematine, or when we describe hematine, it's just a complex phenolic compound that is similar actually to your flavonoids. That is your hematine. So when we say unmordanted hematine, it means po your hematine does not bind easily, or it has poor affinity to the tissue. Okay? So yan po ang ibig sabihin ng unmordanted hematine, class. So this unmordanted hematine in your hematoxylin is actually the one responsible for the yellow color of glycogen in the smear. If ever man na merong glycogen na present sa smear ninyo. Then we proceed to your OG6 or your orange G counter stain. So this one naman po, may number siya na 6 kung mapapansin ninyo. So ano pong dinedenote ng 6 dito? So yung 6 po dito, it actually denotes the concentration of phosphotungstic acid that you have used to formulate OG. Okay, OG. Anyways, so other variants of OG, you have your OG5 and OG8. So again, their numbers differ just because of the concentration of phosphotungstic acid that has been added in the formulation. Okay? So, yun siya. So, si OG po, si OG6, his actual original role was actually to stain the small cells of keratinizing squamous cell carcinoma found in the specimen sputum. Okay? So, yun yung original role niya. Pero ngayon, no, since in-incorporate siya sa Papa Nicolau stain, no, it's commonly used now for gynecological specimens. Okay, and so its role now changed, no, from diagnosing squamous cell carcinoma to uh, diagnosing cervical cancer. Okay, so yun siya. But again, ha, it doesn't mean na hindi na siya pwede gamitin sa squamous cell carcinoma, no? Na madadiagnose through sputum. Pwede pa rin. Kasi remember, di ba, your Pap stain can be used in other specimen types. No? Pero nowadays, it's commonly used dun na sa gynecological smears. No? Kasi remember, Pap stain is most commonly used in gynecological specimens. No? Then the third time is we have our using Azure counter stain or your EA. So this one, we have this what we call EA36. You have also this one we call EA50 and EA65. So as you can see, the number is the only thing that differs here. No? And that number differs from one another. It's because those numbers, no, 36, 50, 65, it just denotes the proportion of the dyes, no, under your Eosin Azure. So ano nga po yung dyes under Eosin Azure? Di ba? Tatlo po yun sila. You have your Eosin Y, your light green SF, and your Bismarck brown Y. Okay? So also, under your Eosin Azure, meron din po siyang, aside from the dyes that I have mentioned, meron din po siyang phosphotungstic acid. And actually, no, even though they are the ingredients, no, needed to form your EA, no, your Bismarck brown and your PTA are actually incompatible. Okay, why? Because they precipitate each other, and since they precipitate each other, no, the useful life of the mixture of your EA counter stain is usually compromised. No, kaya po mapapansin niyo later, no, when we arrive to Bismarck brown, mapapansin niyo later na sasabihin ko na si Bismarck brown hindi na siya usually kasali sa formulation ng Eosin Azure. And you will know another reason later. No, nga, dili naman siya gina-appeal. Okay? Okay, so let's discuss now each of the dyes under your Eosin Azure counter stain. So first is your Eosin Y. So your Eosin Y is the one that stains the superficial epithelial squamous cells, as well as the nucleoli, the cilia, and even the red blood cells, if present in the smear. For the light green SF yellowish naman po, this one stains the cytoplasm of other cells, and actually this includes the non-keratinized squamous cells as well. So your light green SF po, disadvantage lang po nito is that it is expensive, no? And aside from it is expensive, it is also difficult to obtain. That is why there are other manufacturers na aside from, or rather, instead of using light green SF, no, other manufacturers, they switch into using this what we call fast green FCF as their alternative. Okay? But of course, since you have already used another dye, expect po na it could produce, no, a visually different result. And others actually seemed to be dissatisfied with the result. But again, it depends on the expertise, no, of the one reading the smear, as well as the protocol of the laboratory. Okay? And the third dye is your Bismarck brown Y. So this one, as I've said a while ago, since it is incompatible with, what do you call this? Incompatible with the phosphotungstic acid. So as I've said a while ago, it is often omitted in the formulation. But another reason why it is omitted in the formulation, it's because it is useless. Why? How useless? Because it does not stain anything in the smear. No? So why would you place this dye if it does not stain anything? Diba? So kaya po yung recent formulations ng ating Pap stain set, no, tinatanggalan nila yung si Bismarck brown Y as ingredient, no? Okay, so now after you have performed Pap staining, no, it is expected that once you observe it under the microscope, you will observe different hues or different colors, no? And these colors may range from red up until violet. Oh, Roy G. Biv, no? The colors of the rainbow. Also, after doing this staining method, the chromatin patterns are well visible, and the cells from the borderline lesions here are easier to interpret. By the way, what do we mean when we say borderline lesions? When we say borderline lesions, class, these are actually lesions characterized by the presence of atypical or low-grade neoplastic cells. Okay? So it's a lesion characterized by the presence of atypical or low-grade neoplastic cells. So, in addition, when we talk about Papa-Nicolau stain, when you perform it properly, you can actually produce better photomicrographs, and the cells here are very transparent, or should I say translucent. So why do I say so? Kasi po, after performing properly the Pap stain, even though your smears are very thick, and then of course, diba, kapag thick po yung smears, yung cells nyo nag-overlap. So, even though nag-overlap yung cells, thick yung smear mo, you can still actually differentiate them from each other. Okay? So, what do I mean by that? Let me show you a picture. So, this one. So, as you can see, cells here are overlapping, but even though they are overlapping, you can still differentiate one from each other. Okay? So, yan po yung kinagandahan ng Pap stain din. Okay? Now, this one tells us the colors that you will expect and what do these colors mean. Okay? So, yung mga blue to black, like for example ito, blue to black, that's the nucleus of the cell. If may napapansin po kayo na yellow, parehas ito, yellow, so that's actually keratin, ang stain niya keratin. So, ibig sabihin kapag maraming keratin sa isang cell, mapapansin yung entire cell magiging yellow na. So that's actually characterized or described as a keratinized cell. Correct? So also in the smear, if glycogen is present or if in a cell, glycogen is abundant, mag-yellow din po siya. Yung mga superficial cells naman po, they tend to become orange to pink. For your intermediate to parabasal cells, turquoise green to blue, and then for your metaplastic cells, they are often both green and pink. So, if you want to more about these cells, if you want to know more about these cells, I think in your lecture, each of this cell will be described, and it's also in your book, pages 314 to 315, for you to easily locate. Okay? So, yun po yun siya. So, ito superficial cell. Ito yung sila si and then you have your intermediate cell, the endocervical cells, and then you have your metaplastic cell. So, also I would like to tell you, as early as now, so, hindi lang po kayo magbabase lang sa color ng cell, when you try to identify the cells in the smear. Okay? Once you discuss this one in the lecture, you will see na aside from the color, you also need to take note of, for example, the size, the shape of the cell. So, same with how we diagnose, what do you call this? Same with how we diagnose parasitic infections. Diba? We do not only focus on one feature. We also consider other features as well. So, same with your Pap stain now. When you try to read your Pap smear, after staining, you need to consider not only one feature like the color, but you also need to consider other features as well. And I hope you're going to discuss that one in the lecture. So, of course, no matter how good something is, diba? No matter how perfect it might look like, there would always be imperfections. Correct? Wala pong perfect sa mundong ito. Okay? So, there will always be imperfections. So, what could be now the disadvantage? What could be now the problem when using Pap stain? So first, it is not standardized. Okay? So the question is, why is it not standardized? Because po, The formulation here could be different, no? From one manufacturer to another. Other formulations could be different. Also, there are times, no, na the procedure as well, no? Iba-iba, no? In terms sa time, kung gaano siya katagal i-dip, no, sa isang chemical. So, ganun po. No? Nag-iiba po yung timing. Okay? Also, aside from the formulations, there are also modifications. Okay? But actually, no, related lang yan sila, no? Formulations in terms of the chemical and then modifications in terms of the procedure. Okay? Now, let's proceed with a specific, what do you call this, modification, no, for Pap staining. And this is the cytocolor method. So this one is actually a staining kit made by Merck Millipore International. So the staining time, no, for this one, no, when you use this method is only 3 minutes. And it actually requires no orange stain. So sa kanyang procedure po, no, okay, sana ako, it requires no orange stains. Ibig sabihin po, hindi kailangan ng OG6 dito sa method na ito. Okay? So, since wala pong orange stains sa method na ito, so both your mature and keratinized cells, okay, they appear as pink instead of orange. Okay? So, yun siya. So, yung both mature and keratinized cells, they appear as pink na talaga instead of orange. Kasi di ba kanina, doon sa classical na Pap stain, yung inyong keratinized cells, and especially superficial cells na highly keratinized, no, dito po mag-vary yung color niya. Pero since wala ng orange G dito sa cytocolor method, so you expect na pink na ang kanilang color. Okay? So, wala na siyang range. Okay? So, this method, the cytocolor method, actually is based on a specific modification of your Pap smear staining method. And that is the what we call Zepanix, careful sa spelling, Zepanix modification of the classical Pap stain. Okay? So, this one, no, is a cytological staining method used for, again, early recognition of the female genital carcinoma. And no, the results almost same, no? Kasi nga modification na siya ng Pap stain. So, ang results niya hindi masyado ganun ka different, no? Sa anong gusto mong makuha na result from the classical Pap stain. So this modification still gives results regarding the malignancy status of the patient, the hormone status of the patient, and you can still actually observe the vaginal flora of the patient. No? So when you say vaginal flora, you can still observe, no, the microorganisms, no, the bacteria that could be present in the, what they call this, in the smear. Like for example, you want to observe, no, your Lactobacillus acidophilus, no, your Gardnerella vaginalis, no. So those are examples of microorganisms that could be no, present or that you could observe, no, under the Pap smear. Okay, after staining it. Also, no, same with your classical Pap smear staining, no, other specimens could also be used in this modification, such as specimens like specimens from tumors, no, from punctates, from excised specimens, no, from the sediments or the centrifugates from body fluids. Okay, so yun po yun siya. So now, let us proceed with the procedure for Pap smear staining. Okay? Okay? So, let's begin. Wait lang ha, i-zoom natin. Okay. So, these are the materials that you need for Pap smear staining. Okay? And then, I will proceed with the procedure immediately. Okay? Kasi ito yung importante. So, there are two procedures that I would introduce to you now. The first procedure, it will be the traditional or the classic method. And then the second one is the cytocolor method. Okay? So let's begin with the first one. Generally, when we talk about the procedure for Pap smear staining method, it can actually be grouped. No? The steps could actually be grouped into 4. So the first one is fixation. And that's the first one, fixation. The second one is nuclear staining. The third one is cytoplasmic staining. And the fourth one is clearing. Okay? So you can actually group the steps into 4. Again, you have your fixation, nuclear staining, cytoplasmic staining, and clearing. Okay? So what we do here first, we obtain the slide na meron ng smear sa kanya. No? Kasi usually kasi sa hospital class, no, ang gumagawa ng smear ay yung OB-GYN, yung doctor. So pagdating sa laboratory, automatically, na nakasmear na yan siya. Okay? So you obtain first the smear, and then once you have that one, you allow the edges of the smear to dry. And then after nag-dry, no, you can proceed to, you're going to fix the smear in 95% ethanol. O, wala nang ratio dito. Okay? As long as ma-immerse lang yung ating smear sa slide, okay na yan siya. Wala na pong ano dito, wala na pong ratio. Bakit? Kasi we're not talking about tissue blocks here anymore. Okay, so smear na ito siya. So immerse that one for 10 minutes in 95% ethanol. Okay? After that one, okay, after that one, you're going to follow this procedure now. So, first, you're going to, after fixation, you're going to rehydrate. Or rather, hindi siya rehydrate, rather. Hydrate pa muna. Now, you're going to hydrate. Hindi siya rehydrate kasi hindi man kayo nag-hydrate kanina. So, you're going first to hydrate the cells. Okay, kaya pa mapapansin niyo, di ba? Decreasing ang grades of alcohol. Okay? So, you're going to hydrate. Sir, bakit kailangan mag-hydrate ng cells? No? Bakit kailangan i-hydrate ang smear? This is to prepare for hematoxylin immersion later on. Okay? So, also, if you can see, no, varying grades pa rin, no, of alcohol, varying, decreasing grades of alcohol pa rin. Kasi again, we're still trying to minimize cell distortion as well as cell loss from the glass slide. Okay? So, after the decreasing grades of alcohol, you're going to rinse it with distilled water, and then you're going to place it or immerse it in hematoxylin. Harris hematoxylin. So what is the purpose of your Harris hematoxylin? It is for nuclear staining. Okay? Then you rinse it with distilled water. Okay? Then you proceed to diluted hydrochloric acid. Diluted na siya, okay, 0.5% naman. So you proceed to diluted hydrochloric acid. The purpose of your diluted hydrochloric acid is still to remove, no? It is to remove the excess hematoxylin. Okay, that is still the purpose. So same siya with the the function of distilled water. So yung mga hindi natanggal ni distilled water, na mga excess na hematoxylin na stain, magtatanggal niyan ay si diluted na hydrochloric acid. And then, distilled water ulit, no, para ma-rinse yung ating smear from hydrochloric acid. And then after distilled water, we proceed with our bluing agent. So meron din pong bluing agent, si classical method. So the bluing agent here is ammonium hydroxide. Okay? So, what is the purpose of the bluing agent here? Now, what is the purpose of ammonium hydroxide? The purpose of ammonium hydroxide here, class, is to subject our hematoxylin in alkaline condition. And as a consequence of that one, the color now of your hematoxylin stained the mga cells, it will become, from red, it will become blue. Okay? So, specifically, anong part ng cell ang magre-red to blue? It's the nucleus. Kasi diba, hematoxylin, no? Yung ating subject to alkaline condition here. And again, it is a nuclear stain, no? Si hematoxylin. Okay? So, ang mangyayari dito, after natin ma-immerse in ammonium hydroxide, yung nucleus, no, ng cell, from red, magiging blue siya. Sir, gaano katagal ang ating pag-immerse or ating pag-dip sa ammonium hydroxide? Hanggang sa may ma-observe na kayo na tinge of blue. Okay? Now, after subjecting it to ammonium hydroxide, you proceed now to the increasing grades of alcohol. So, obviously, increasing na po grades yung alcohol dito. So, ang main goal natin is to dehydrate now again the cells. So, not again, rather to dehydrate. No, hindi ako sa again ngayon. Anyways, to dehydrate the cells. Okay, so that's the purpose. So to dehydrate the cells. Okay, after dehydration, no, after immersing it to increasing grades of ethanol, you subject it to orange G6. Or you immerse it in orange G6. So your OG6, this one now is a cytoplasmic stain. No? But this stain class is actually just a monochromatic stain. Ibig sabihin po, isang color lang yung kanyang pwedeng i-contribute sa smear. And anong color yon? Obviously, it's orange. So since orange lang yung ma-contribute, and also if you're going to relate it to the concepts I have discussed a while ago, anong klaseng cells lang yung mabibigyan niya ng color? Anong cytoplasm? Rather, anong cells? Anong cells ang mabibigyan niya ng color? Specifically, anong cytoplasm na nabebelong? Bibigyan ng color dito. It's the cytoplasm of your keratinized cells. Okay? Now, after subjecting it to OG6, no, you immerse it to 95% ethyl alcohol. So the purpose of this one, this 95% ethyl alcohol, is to rinse our OG6. Then after rinsing, we proceed with EA65. So your EA65 is also a cytoplasmic stain. But this one, it is already capable of inducing different colors, not only orange like your OG6, but this one can also can now induce or produce, mas appropriate in term na produce different colors that you can now observe under the smear. Okay, then after immersing it to EA65, you again immerse it to 95% ethyl alcohol. So the purpose of 95% ethanol here is for rinsing again. And then after 95%, we have your pure, your absolute ethanol. So the purpose now of your absolute ethanol is to dehydrate completely the smear. Okay, to dehydrate completely the smear. And then the last step is silane. So obviously, the purpose of silane here is for clearing. Okay, so after silane, after clearing, you proceed to mounting. Okay, so that is the procedure for the classical method of pap staining. Now let's proceed with the cytocolor method. Okay, the cytocolor method. Okay, so for the cytocolor method, it's quite shorter in terms of time and in terms of the number of steps. No, kaya po, di ba, sabi ko kanina, sa cytocolor method, ang kanyang staining time lang is 3 minutes. Di ba? So, konti na lang po yung steps dito. So, nandyan pa rin po si fixation sa cytocolor method. Nandyan pa rin yung fixation with 95% ethanol. Nandyan pa rin yung rinsing with distilled water. Of course, nandyan pa rin po si hematoxylin, which is our nuclear stain. And then rinsing again. And then this time, we use 2-propanol for dehydration. Okay. And then we have a polychromic solution. So this one is the cytoplasmic stain. And then we proceed to 80% 2-propanol. So this one is again for rinsing. And then the 2-propanols here are for dehydration. And then finally, we still end with clearing with your silene. And then to preserve the slide or the smear, mounting. Okay? So those are the steps for the Cytocolor method. And that will be the end of the discussion for Pap smear staining. So thank you for listening.